So today we're going to talk about physics of light, which is important to understand before we start talking about photosynthesis. And then our next topic will be a few slides on the chemiosmotic production of ATP before we actually start talking about the chemiosmotic production of ATP. And then we'll talk about the light reactions, the dark reactions, then go into metabolism. Then I'm probably going to push up secondary metabolism, so we're going to have probably more than one lecture on secondary metabolism. That's becoming more and more important. As you know, we're finding more plant-based whatever, drugs, different uses for plants. That's becoming more of an important thing. And then we will do a little bit of growth and development and then do some hormones. Now, here's what we're going to be talking about. Understand the physics of absorption. Understand the nature of light. I'm not going to go over these. You know, this is like the basic physics of how light is so weird. You know, because light is both a particle and a wavelength. And I think maybe other than Einstein, really no one really understands what light is, maybe. And then we're going to talk about how absorption occurs and then talk about the pigments. Now, these are pretty obvious. We're looking at photosynthesis, and that specific area is called photobiology. And this is a really hot area even now. DARPA funds, anybody who knows what DARPA is? The defense research. You know, they would love to figure out how we could use photosynthesis as a way of producing energy, you know, artificially. That would be fantastic. So DARPA funds a lot of work on photobiology. And let me tell you now, we don't know everything about photosynthesis. Now, what is light? Light energy is everything that's falling, it's coming out of these bulbs, that's coming through the walls that we can't see. If we could see a really broad spectrum, everything would be just like bright white because we'd be seeing so much energy. We only see a very little bit of the energy. And then radiation is the term we use to describe these different regions. And got a couple of these. So if we look at the actual spectrum, we have the famous equation, which you'll see. C is equal to nu lambda. So we have increasing frequency is absolutely the opposite of increasing wavelength. So shorter the wavelength, the higher the frequency. And you know that, you know, gamma rays are not a good thing to absorb. UV, if we absorb UV, what happens? Causes thymine dimers, and your cells then try to repair that, and we call that sunburn. So when you're actually getting a sunburn, you're actually damaging your DNA. And then your body goes through a lot of inflammation, which is a big thing that medical people are looking at now because inflammation is very important in terms of disease process. And we do absorb, unfortunately, some UV. Also, just the absorption of the energy can help damage membranes. And one thing I want to keep getting across is that light has to be absorbed for it to do anything. So when light is absorbed, in the case of photosynthesis, that energy, that actual photon moves through a lot of reactions, and that photon, that electron ends up reducing compounds. But if light is absorbed, it can do several different things, and we'll talk about those. It could be given off as heat. It can be given off as a different kind of particle, but we don't really know when this does not have much effect on us. This is a little bit of what we see. Some insects see a little bit into the UV, and I think when I do secondary metabolism, I have a neat picture of a flower that we see, and then a flower that an insect sees, and it looks just like a bullseye. Yes, do you have a question?
Yeah. What about plant DNA allows them to like repair itself or not be affected by like UV?
Why don't plants get sunburned? Because the pigments that are in the plants. Okay.
Because I mean, they just sit out in the sun and just get roasted.
Right, and we can have some horticultural problems. We can have sun scald in tomato, and tomatoes grow up under the leaves, right? So if you pick a lot of tomatoes in the middle of the summer in Texas and you set them at the end of the road, they're absorbing light energy, and they can't do anything with that light energy. So we do have damage. Okay, but they're basically designed because they've got to be out all the time.
Yeah.
So they can actually make pigments. Plants can actually make pigments in response to light, mostly UV. It's a brand new thing now is that if you expose plants to UV light, they may start producing a red anthocyanin pigment. There are, if we look back at when evolution started, we started out with green things in the water, so the water absorbs a lot of the energy. And if we look at what, let's say a red algae absorbs, red is what plants don't absorb that well, you know. And there's some algae that look black because they absorb almost all the radiation. Okay. So just to give you an idea of the amount of energy, that we see a decrease here as we get up in the infrared. And this is from your book. And this is just another diagram of the different kinds of light. So we have solar rays, we have sound way down here, radio waves, infrared, and then this little tiny bit in the center here, which is blown out here. And just to show you these curves, which you're going to see over and over again, these are absorbance curves. So we do absorbance by shining the light on a sample, and then we figure out how much light comes through the sample on the other side. And so you'll see chlorophyll A and chlorophyll B, and these compounds have to absorb to do work. Here's the carotenoids. So you see they're absorbing, you know, between 400 and 500 and then up into the 700s. Now, okay, this is the offshoot of a C equals nu lambda. Anybody remember who came up with this equation? You all should know this cold. Probably the most famous equation. Einstein. This was what he came up with. That is, what's C? Speed of light. It's a constant. That was the big thing. No matter what, light is the same speed, which means if one of these terms goes up, the other term has to go down, right? So, if increasing frequency, increase in frequency, the wavelength has to go down. That is what Einstein basically came up with. And so we have this nature of light in which we can measure light radiation. There's a wavelength, so these lights are giving off some, not really white light, but sort of yellowish colored light probably. And at the same time, that wave is also a particle. So not only when we talk about wavelengths of light, we can actually talk about a mole of light, a quantity of light. And you will, if you take Matt Kent's course, he talks a lot about, interior scape talks about the quantity of light, not just the kind of light, but we can actually measure an amount of light. So we have a wavelength and a particle, and it's the same thing. No, it's light, energy. Now, what we talk about a particular kind of particle, we call it a photon. Now, I'm going to use the term photon and electron more or less interchangeably. They're not the same. But in terms of what we're trying to discuss, we're going to talk about a photon of energy, and we're going to need several photons of energy to end up with an electron, and that electron is going to reduce something by Friday that ends up then turning the light photon into chemical energy. So that's what photosynthesis does, is we harvest light photons and turn them into electrons, which reduce things, okay? And then the energy of a particular photon, the wavelength, is called a quanta, and you don't have to worry about the equation. This, you've probably heard of Planck's constant. So what is absorbance? What actually happens? What has to happen when a photon is absorbed? First of all, it's got to be able to occupy a space that is just right for that wavelength and that kind of light. So most light is not absorbed. That's why we see each other. Okay? So we see things because light is reflected. Reflection means the light is bouncing off. So if you think about it down to the atomic level, you've got that photon coming in, and it has to be right in the right place to excite. Remember the excited states, and we're not going to go into triplets or singlets or anything. But it has to be a very specific wavelength. And you probably remember this, and I put this here, which is not correct, not absolutely correct, but it's good enough. And you probably remember this from chemistry or from physics, where a photon then is able to add its energy and it excites an electron, okay? So you have a happy little molecule or an atom in a molecule, and now you've got an excited electron. And we know that we have an excited electron, all sorts of things can happen. And basically, in photosynthesis, we're moving that electron through a lot of steps, as you'll see, to end up harvesting this energy as electrons. So what we're talking about in photosynthesis is doing photochemistry, which means we're driving a series, and like I've told you over and over again, life is basically a series of oxidation and reduction reactions. And where does it start? In the case of plants, that electron, which will reduce, comes from light energy. And these things that absorb light are, of course, called pigments. And different pigments absorb light and reflect other light. Now, let's talk about what can happen. We'll talk about entropy a little bit next time, just a little tiny bit. But what happens when light is absorbed? Well, or when a photon is absorbed? It can begin off as heat, remember? Everything is, we're trying desperately to move to entropy, but for reactions to occur, we are the opposite of entropy. Entropy is randomness. So another thing, and I'll show this to you, is fluorescence. And that's the basic, I should have brought my UV light down, but you've all seen, you shine a a UV light and things are bright. So what you're seeing is absorbance of the light and readmitted at a longer wavelength. So the UV light you really technically can't see, and it's absorbing and emitted. It's got to be a longer wavelength. It can't be the same amount of energy. So that's why you have UV lights and things light up. And I'll bring my lights down next time. Now, the thing that we are really interested in is something called inductive resonance or radiationless transfer. What that means is that energy is going to move from molecule to molecule, from around atom to atom. And that energy is eventually going to end up reducing things.
Is that why it bounces around the complexes?
Yes.
When it goes through the Z-scheme?
Yeah, when we go through that, that's exactly what's happening. It's the electrons are moving around until they get to the correct place, right? And then something that can also happen is photooxidation. Photooxidation is something that can occur in a tomato fruit if it absorbs too much light. You have oxidation, you have ripping of membranes, you can get creams that stop oxidation in your skin. Oxidation, remember, is a big thing that people talk about now. Antioxidants, that kind of stuff. So you can have some damage done in addition just to having your DNA damaged, And think about it on a grand scale. Think about having a black T-shirt on in the middle of the summer in Texas. You get hot. The energy's being absorbed. It's being readmitted as heat. But think about if your black T-shirt had membranes in it, then those membranes could be damaged. So you can have oxidation occurring. Okay? So these are several of the things, not the only thing that can happen. And we show this in a diagram by looking at energy level. Here we have two different wavelengths of light. And don't worry about, if you may remember S1 and S2. So you can have these excited states, and then we can lose heat. F is for fluorescence. P is something we've not really talked about, is phosphorescence. But basically, most of the time, if we're not talking about photosynthesis, that absorbed photon is going to be readmitted in some of different ways. It's not going to do anything. Okay, so only chlorophyll, and other pigments, are capable of channeling that energy and doing something with it. Not just chlorophyll, other pigments, okay? And then this little schematic at the right just shows you where the absorbance is occurring here. So this is just something you may remember from physics. Phosphorescence, okay? Now, what do these molecules have in common? And don't say carbon. What do they have in common? They're all pigments. No. What structurally do they have in common? Well, maybe you can't see. Yeah, you can. Look at the bonds. What are the bonds? What's so weird about the bonding between the ends there? Can you see that? Yeah, if you look at these, maybe you can't see that. Can you see there's a double bond, a single bond, a double bond, a single bond, a double bond, a single bond, a double bond, a single bond? So you guys remember bonds flipping? These things absorb energy, and they also photons, and they transfer it by resonance. So let's say, and this isn't exactly the way it happens, but that's okay. We have something absorbed here, and then it moves this to a double bond, which then becomes a double bond. And so we're able to, these very specific molecules are able to take these photons and move them around. And what you're going to see is what photosynthesis is, the pigments, is that they're arranged almost like a parabolic, like one of the antennas you see from dish TV. They absorb energy and they concentrate it into the center, okay? Why do you have that dish? You know, what's the purpose of the dish that you've got if you have whatever, TV? What's the purpose of the dish? Why is it shaped like that? What's it do?
Whether it hits like out here.
Yeah. In the case of a dish TV antenna, you've got the little receiver above that, the dish. And so the radiation hits and then bounces up to the collector, okay? Photosynthesis is exactly like that, except the collector's down inside, okay? So you're concentrating that energy into one particular place. And that's what photosynthesis is. You're taking this energy and you're moving it into one specific place, okay? You're concentrating it. These are the pigments that absorb. This is chlorophyll, and these are the carotenes or carotenoids that are very important to photosynthesis. Now, so how do we know what does what? Light has to be absorbed, and in the case of photosynthesis, we can measure several different things. So if photosynthesis is happening, what can we measure? When you guys know, we measure oxygen uptake, oxygen evolution. We can measure CO2 uptake. We know that about photosynthesis. We've known that since Priestley, who's the guy that discovered oxygen. That we've known plants produce gas or give off gas and whatever. And so we do something that's fairly important, is we not only plot where the pigment is absorbed, or the electrons are absorbed, photons are absorbed, but is there an action? Okay? So what we do then is we take a plant and we isolate all the pigments in it. We're pretty sure it's green is going to be the chlorophyll. We think that's what's happening, right? So in this bottom curve, and this is very important, you see what happens when you put chlorophyll in a cuvette, put it in a speck, and shoot light through it at different wavelengths and see what's absorbed. A cheap spectrophotometer, it's just a prism. What's a prism do? It splits light into different wavelengths, and so you can, when you turn the knob on like a Spec 20, you're actually aiming the prism. You're changing the prism around. And then you see what goes through the sample and what's absorbed. So what we see is that we have light that is absorbed a little bit above 400 and then between 650 and 700. Okay, that's chlorophyll. The important thing is, then we take our plant and we expose that plant to the same different wavelengths of light, and we measure, and write this down, a biological action. And in this case, we are measuring relative rate of photosynthesis, whatever that is. So we can measure oxygen evolution, or we can measure CO2 uptake. And so what we see is curve A. So if the pigment is absorbing here maximally, we also get, let's say, the highest rate of oxygen evolution, oxygen production. Then if we go down to where there's very little absorption, then we drop in how much oxygen is being produced. And then if we take it up to 680, we see the second peak in oxygen production. So this is an action spectrum. It's an action. So now we have some data that associates the production of oxygen at the same wavelengths that chlorophyll absorbs light. So now we have some good data that indicates that maybe chlorophyll is what is causing this. That makes sense? And you can do an action spectrum for seed germination. Now, what different wavelengths isolate pigments from seeds, and we'll talk about phytochrome and other pigments later, and see which pigment you can associate with a biological action. That's what's called an action. It's a biological action spectrum. Okay? Questions about that? So we're taking all of these molecules and we're moving them. These are very complexly arranged, and we're taking the photon, and at some point the photon becomes an electron, or the multiple photons, and we're moving them around, and we're eventually ending up with these electrons reducing compounds. Okay? Questions before we start talking about what we're actually measuring? So let's talk about some terms that are important in terms of how we measure, like quantity. Okay, that. What about quality? You know, these are they incandescent? I don't know. You know, different kinds of lights are going to give you certain different kinds of wavelengths of light. So probably can't grow plants really well, even if these lights in here were at a very high intensity. You know, because for the plant to make as much oxygen as it can, or give off as much oxygen as it can, or make CO2, fix CO2, you need more wavelengths than these, I guess, incandescent lights. So you look at them, you can tell they're yellow. Okay? So we have light quality, we have the spectral composition, or the spectral energy distribution. Kind of light, how much light, quantity of light, how much, the kind of light, and then, of course, how long the light. So, more light you have, higher intensity light at the wavelengths that we need, which are 700 and 600 or 400, whatever. For a longer period of time, we're going to have more carbon fixed. So we're going to have more CO2 fixed. Okay? So, if we go back and we look at the energy distribution, you can look at the fluence rate of the photon or the energy fluence. And another term you'll see is spectral irradiation. Okay? So the SEDs, these values, does Matt talk about SED, or does he use anybody had interior scape with Matt Kent? I'll have to ask him. And so now we get to the take-home message is what we're actually seeing hit, in this case, what is outside the atmosphere, what makes it into the atmosphere, and then if we look at the intensity of a fluorescence versus an incandescent light. Okay? So, and you'll see this, you don't see this as much around here, but the further up north you move, the more supplemental lighting you'll have even in a greenhouse. And you can't just put light bulbs, you've got to get a mix of different kinds of light. You can get grow lights, you can get very specialized lights. The big thing now is LEDs, and I don't think that's right, not, In the lab, the next corridor, if you go by, there's some racks with a lot of lights. And I don't, is that in the 202 lab? They're all LEDs. LEDs are really coming on strong because with LEDs, we can, in some cases, produce any wavelength you want. They're expensive. They last forever. You can actually get a set of lights from, oh goodness, I can't remember. I want to say you can buy them through Apple, but that are not made by Apple. There are 3 LED lights. They're about $200. And what's really weird is they will produce any wavelength that you can see. And you can get an app that is tied into the, I can't remember what project it is, where they're identifying new planets. You know, it's pretty, I mean, it's common now that we identify planets, you know, in other systems. Well, if you're really a geek, you can get this app, and so you can be sitting at home with these lights, and if a new planet is found, it plugs in the information, and the lights change to the spectra that planet sees. Okay? I can't remember who makes the lights. They're becoming more and more common.
Pluto's still a planet, though.
I'm sorry?
Pluto is still a planet.
Pluto? No, it's not.
Well, I'll argue about that.
Okay, so now let's talk back about biology. Photoreceptors, pretty much what it says. And we can have a lot of different kind of photoreceptors. A chromoprotein, that's a protein that's colored, that can actually maybe absorb some light. And it consists of a pigment, which is the chromophore, and then a protein, which is the apoprotein. And don't worry much about that. Just the point is there are things that can respond to light energy. And now we get to the one everybody knows about, chlorophyll, which has a porphyrin ring, which is the ring structure, and a phytotail. And the phytotail, and don't worry about this, is isoprene, just that it has a long hydrocarbon tail, and magnesium is needed in the middle. And I think I said iron was needed in a lecture. I made a mistake. I think I said iron. And so we have this molecule. Now, just looking at this molecule, where would you find it? You know it's in the chloroplast, but where would you find it? Based on the solubility of this molecule, where do you think you would find it? In the membranes. It's got this long hydrocarbon tail, right? And it's not very water soluble. Chlorophyll is not water soluble. But just structurally you look at it and you can see, well, this looks like something I'd find in a membrane. And of course it is. And so like I've told you over and over and over again, when you look at a tree that's green, the leaf is not green, the chloroplast is not green. It is the chlorophyll molecules in the membrane, in the inner chlorophyll, not mitochondria, the inner chlorophyll membrane, that's actually the little green dots. So if you could blow up a chloroplast and look at it, you'd be able to see dots of green and yellow, because carotenoids are also incredibly important. So when you see a green leaf, you're actually seeing the chlorophyll molecules in the membranes. And membranes, for the most part, are clear. They don't absorb much of anything. Okay? And here we have chlorophyll B and chlorophyll A. You don't have to worry about the difference between the two. It's just there are two chlorophylls. They're extremely important in plants, the ratio of chlorophyll A to chlorophyll B. And that will vary from species to species or from algae to different kinds of plants. Now let's talk about something that you may not be as familiar with, which is the carotenoids. And these are incredibly important. because they have that same double single lipid soluble structure, and they absorb a tremendous amount of photons and channel it into the site where we actually have photochemistry occurring. And carotenoids are also found in chromoplast. Where's probably the most common chromoplast you see? When you go into a supermarket, what fruit has the chromoplast? Tomatoes. So the red that you're seeing in a tomato is carotenoids, and that's one of the main reasons why tomatoes will get sunburned. They're actually absorbing that energy. They can't do anything with it. Okay? There are carotenes and xanthophylls, and they are very important. Just to give you, you don't need to know this, in addition to absorbing blue light, beta-carotene quenches triplet-excited chlorophyll, protecting it from photooxidation. We will talk a little bit about what happens when you have too much light, and too much light can damage a plant. And one of the things that carotene can do is help keep that from happening. It can channel off photons or electrons, whatever you want to call it. Here again are these same structures. You look at these structures and say, I think I'm going to find this in a membrane, right? And they absorb at slightly different wavelengths. So if we look at them together, here is the chlorophylls and here are the carotenoids. So they're absorbing only down here as opposed to where chlorophylls also absorb up here in the red, far red, which is very important. And then the last one we'll talk about, next to the last one, are flavonoids. And this is what causes plants to be colored. Red, whatever, different colors. These are non-photosynthetic pigments. Most of these are water-soluble, so where are you going to find them? In the vacuole. That's why vacuoles, and I think I've got a picture of why vacuoles are colored. And they're very important. They have become really important in the last few years as antioxidants. So, you know, if you drink red wine, you'll never die. Or if you eat a lot of vegetables that are high in antioxidants, then, you know, you're never going to get cancer. All this other BS that you hear. And they're very important. And this basically is what they look like. These little, smaller molecules. And look at here, it doesn't have a long chain on it, so it's probably going to be water soluble. And they tend to absorb, and I tried to set these off, so here's 300 and here's 300. And they tend to absorb in an area that's not needed for photosynthesis. Obviously, if they absorbed at the same place, photosynthesis would not be as efficient, would it? Because energy would be absorbed for this by these molecules. And here is, just to give you an idea, here is a, I don't know what kind of, probably LED or whatever, but you can see in this case the vacuoles are red. And then we want to finish up with this one, Betalins, which you may or may have not heard of. And this is well, it's been around forever, but it's the newest one that's been found. It's been around for a long time, but it was debated whether or not it was an actual flavonoid or not. And this is another kind of compound. The big difference here is that these things contain nitrogen, and this is the red you find in beets. And a few other plants, okay? Totally different kind of compound, water soluble. It's easy to tell if a pigment is water soluble if you eat it or if you cook it. If you cook carrots, the water doesn't turn bright orange, does it? If you go look at beets, the water is bright purple. That tells you whether or not it's water soluble or not. Asparagus, the liquid is sort of green, but it's not as green as the asparagus, okay? Questions? Okay, that's the lecture for today, and I'll see you on Wednesday.